Thank you very much for allowing me to unmute. Uh, thank you, Dr. Sikandar Azam, for giving a scientific presentation. And this type of a study is, I think, very important to understand the mechanism of action of the virus, the response of the human cell. And this is a very valuable study to move forward to understand and to overcome this pandemic. Our next speaker, Dr. Vass, I would also like you, because we are running short of time, to be brief and remain within 10 minutes. Dr. Vass is a graduate. He did his degree in biotechnology from Qaid University, and then did his PhD at Chinese Academy of Sciences. And his major work is on nanomaterials and nano safety. Based on technological development, is also a co-founder of a startup, Synthon Nanotech headquartered in the Netherlands, which aims to develop assembled short peptide-based nanomaterials for cancer, Theranostics, antimicrobial application, 3D cells culture, and bioprinting. And the topic of his discussion is now on our screen. Now I invite Dr. Vass to deliver his talk. Thank you very much. Over to Dr. Vass. Dr. Vass, we can't hear you at the moment. Can you please? Dr. Vass, can you hear me? I think we can ask the host to unmute Dr. Vass, because he might be having a problem with unmuting his mic.
Hello, can you hear me now?
Yes, we can hear you now. Go ahead.
Okay,
please.
thank you. All right, so today, thank you so much, the organizing committee, for giving me the opportunity to give a talk on the nanotechnologies for COVID theranostics. So before going into the detail of this topic, I will clarify the word like the theranostics, number one. You know, like the nanotechnologies have been used for different diseases in terms of its diagnosis and therapy. So today's topic, I will focus on the COVID-19 and how the nanotechnologies have been focused on the COVID-19 in terms of its diagnosis and therapy. So, you know, not only the diagnostic perspective of the nanotechnology or the therapeutic perspective of nanotechnology, nanotechnologies have been broadly used, you know, since 1900s, like from 1960s onwards, actually going into the market from the 2000s, and then currently we can see that there are a lot of applications involved. So in terms of COVID-19, in terms of the viruses, that's not only been used in terms of vaccines and you know, in terms of the therapeutics, but also for the masks, the clothes, and many other applications. So coming towards the nanotechnology for COVID-19 therapeutics and vaccine research, as we know that we are, you know, passing through a difficult phase of COVID-19. So the there are, you know, scientists are working on, you know, developing new therapeutics and exploring new biomolecules so that they can, you know, counter the disease of COVID-19. So how the nanotechnologies is helpful in this case, and actually what is the nanotechnology in terms of its biomaterial potential. So for the layman in this who are not familiar with the nanotechnologies background, you know, I will just give you an overview that in the nanotechnology we use nanoparticles that are at the size of 10 power to minus 9 nanometers. So they're very minute and we cannot see them, you know, from the naked eye. And these particles actually have the potential to help us diagnose and treat the cancers. So we call it like the viruses are also the nano enemies, and the particles we are using are also nano in nature. So how the strategy works is like a bit, this presentation will be a bit more technical, a bit more clinical, but I will talk more on the interface of the nanotechnology and the immune cells and how this all interface works. So primarily the blocking the the interaction of the viral spike glycoprotein with the host surface cell receptors and disruption of the viral construction. So that's how it works. This is a pretty complex figure, as you can see, but it's not that much complex if you understand it. The point is that, as you can see in the left side, that's the uh, endothelial layer of the alveolar macrophages. So and that's the endothelial one, and that's the epithelial one. So when the in the airways, so when the actually the virus attacks. So in this figure, what I discussed is about the healthy and dysfunctional immune response during the COVID-19. So infection. So when the virus attacks, the cell goes into the paraptosis phase, and there are a lot of immunological interactions are being carried out within the within the immune cells. So there are two ways in which it can lead. Either it can go towards the healthy immune response, so as you can see in the green one, or it can go to the dysfunctional immune response. So when it goes, there's a lot of details involved in it, but I will not go into it. But the point is that when it's like a healthy immune response, it's very evident that the virus is being cleared up from the from the lungs. And but if it is like the dysfunction of the immune response, so it's not being cleared up. So from this slide, it's like what we have to understand is that it's very important for us to know that how strong immune system should be there, and the immune system by its side, that how the nanotechnologies can be used to actually strengthen the immune system of ours, to fight against the disease. So these are in this slide you can see the existing antiviral molecules that are being under developed for the COVID-19. So they are like from the 1 to the 9, you can see the phase how the virus works. So it starts from the CoV2 E2 binding, right from the virus release till the 9. So these, in all these stages, we have different biomolecules. I have different drugs that are being, like, they're already antivirals, and they are being repurposed for against COVID-19, starting from the remdesivir, gladstevir, tenofovir, and flepivir, and different one. So from the proteolytic cleavage, the viral release, translation, transcription, all these things. So now, till now, we have cleared about that there are been drugs which are being repurposed for the COVID-19. So here, coming towards my core discussion of the nanomedicines, that like how the COVID-19 nanomedicines, you know, uh, 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 helps into the vaccine developmental efficacy on one side and addressing the therapeutic one. So therapeutic, the upper one, this portion, the upper portion is like therapeutic one, how how the nanotechnologies are being helpful in that purpose, and the vaccine one. So currently, as we know, that the vaccine has been developed by the Moderna company in the USA, and the BioNTech, they are the mRNA-based vaccines. And actually, it's very interesting that those mRNA are being delivered with the help of lipid nanoparticles. So the nanotechnology in today's time is of pertinent. It's so very important to so that we can do further research in that. That actually the vaccines we are getting in our the Moderna and the BioNTech one, they are actually the nanoparticles, and those nanoparticles have incorporated the mRNA. Then there are different types of vaccines. As we know, that mRNA is one, and then you have the conventional one. So in this slide, you can see that there are different ways that how therapeutics are made in terms of nanomedicine, with the combination, with the repurposing, and the chemical alteration. And in the vaccine side, we can see that how this mean made. For example, antigen dependent nanocarrier selection. So that's what I talk about the Moderna and BioNTech vaccines. Okay, so there are different types of carriers which we can use. Like there are like the liposomes, which I talk about, which has been used by and the combination of liposomes and polymeric has been actually used by the with the current vaccines. But there are a lot of other particles which can be used, like the dendrimers, the mesophorus silica nanoparticles, core shell, layer by layer, and all these things. So in this slide, you can see that it's like this slide shows the interface of how the nanoparticles actually incorporating the antigen interacts with the immune cells. So it's like a it's like a very clinical, but it's very easy to understand. Like the nanoparticles, as you can see on the left side in the A, delivery of antigens. So the antigen delivery actually depends on the size of the nanoparticle. So the cells which we are targeting, for example, if you are targeting a macrophage, if you are targeting a CD8 T cell, whatever cell we are targeting, we have to design our nanoparticles as per that particular cell size so that it can go and directly targets it. Secondly, it's important to talk about the dapper effect in terms of the nanotechnology and how it's been used for the COVID-19 theranostics. So in the dapper effect, we make a reservoir of the antigens incorporated inside into the nanoparticles, and these reservoirs sustain release actually the antigens, so that those antigens are being then taken up by the macrophages and the immune cells, and then they show them on their on their surface on the cell membrane, and triggers the immune response against them. So that's how it works. And then we have the repetitive antigen display and the cross penetration. There's all the mechanisms involved. This slide shows the major delivery ways for the mRNA and DNA vaccines. So on the top one, as you can see, you see the cholesterol, right? And the PEG lipid. So the lipid nanoparticles are being made like this. So it's like a lipid bilayer, and that includes the mRNA or our therapeutic targets. And then you can see from the 1 to 5, these are all carriers being designed in a way to transfer mRNA, which are currently being in the market. So furthermore, from 6 and 7 1, as you can see, this is for the DNA one. So that how is the DNA molecules are being integrated into a single PLGA nanoparticle. So the glycol, chitosan, polyethylenamine. So there are different sort of molecules which the nanoscientists which we play with, and we do this. We make these nanocarriers. So in this slide, you can see it's like a paper which I have published with my collaborators. It's talk about the phage capsid nanoparticles as multivalent inhibitors of viral infection. So here we have given an idea that designing a multivalent nanoparticle will be really good in terms of its therapeutic potential as compared to a single valent or a simple nanoparticle. So the multivalent nanoparticles, as you can see in the figure here, like they have they attach with the spike protein, and they have the more potential to be specified in terms of COVID-19. And there was also a paper published in 2020 in Nature Nanotech, which they have used the same nanoparticles for the influenza virus. Giving the potential of the nanotechnology in terms of the therapeutics and also in terms of the diagnosis. And this is a paper which I will recommend the researchers to read, is about that how this actual detection of the COVID-19 works with the help of nanotechnologies. So here you can see is a schematic diagram of the COVID-19 FET sensor operational procedure. So graphene material is a very famous material in terms of our field of nanotechnology, is being used here, and the SARS-CoV-2 spike antibodies have been conjugated in here, on the FET sensor. So when the source of SARS-CoV is in there, where the sample is entered, so the sample attaches with the spike antibodies, and then we can have a signal out there so that we can detect it. So there is a global innovation being happening at a very high pace in terms of both diagnosis and therapy of the COVID-19 with the help of nanotechnologies. And these examples which I have shared are just the few of the examples. There is a lot of diversity in there, a lot of new techniques, a lot of new ways to counter and use this nanotechnology for the COVID-19. So coming to the conclusions of my today's presentation, the nanotechnology scientists actually, they have diverse backgrounds. So that they come from the chemistry, physics, and biology. But it's very important for all of us working in the COVID-19 domain to actually understand the pathophysiology and the immunological response mechanisms, so that we can actually make a very, do important contribution in terms of the COVID-19 theranostics. And then obviously, it's very important to make more effective vaccines vaccine strategies. Recently, we hear about the COVID variants, coming out in different countries. There are studies which shows that obviously the Moderna and the other vaccines which are being in the market are being actually are helpful against some variants, but maybe not. And there is need of further studies so that we can know that which vaccines are working against which variants, so that because the virus is multiplying at very high pace. And lastly, the delivery and of effective biomolecules like the mRNAs are very important, as we can see in currently the vaccines in the market. They're all made up of the mRNA and the nanoparticles. So it's very important to for us to understand this mechanism and further to direct our research on this strategy as well. So thank you so much, and thanks for listening.